Welcome everybody to the meeting. Thank you for joining us. And I would like to call this meeting to order. First thing, Secretary Gary Havel, could you please read the minutes from the last regular meeting?
Yes. The 1257th regular meeting of the Entomological Society of Washington was held at 7 p.m. on January the 8th, 2026 in room WG 33 of the National Museum of Natural History. Present were 20 members, 6 guests, and 18 virtual participants. The meeting began with a reading of the previous meeting minutes by Recording Secretary Gary Havel, with approval by members of the meeting at the meeting. Membership and Communications Secretary Matt Buffington noted several applications for society membership. Carson Kane, Don Ferschau, Freshacil, John Hash, David Davis, Allred, Vernon Brew, Zach Lahey, and Steve Gameri. Curator Alyssa Seaman announced the technical upgrade of the society microphone to enhance communication. Frank Krell introduced himself as a new research scientist with APHIS, noting that he was hatched in Germany and has been at the Denver Museum for the past 18 years. His specialty is Scarabaeidae. He also noted that he has agreed to serve as a subject editor for the proceedings. David Firth noted the recent visit by Mike Farrell to transport entomological literature from the museum to Clemson University, where the reprints and books will be made available to entomologists with special interest in them. David also noted that he has recently been to the Dave Adamski house to clear various entomological items. He noted that among the material were original color illustrations of serpentine leaf miners by Snodgrass done in 1920. Dave mentioned that Tom Eisner had said that the best time of his life was at the Smithsonian with Snodgrass. Unidentified person, Rose, I understand, got to get that name, displayed knitted items of insects done by her friend Meredith Wilmot of Falls Church. Don Weber noted that the ESA is changing bylaws and offered information on this to anyone interested in the changes. Program Chair Renan Carinho arranged the speaker of the evening, Catherine Malinsky. Ted Schultz introduced her, noting that she was currently working with him at the museum. She received her doctoral degree in 2024 and has joined many workers in the fascinating field of fungus-feeding ant research. She and her colleagues are concentrating on fungus-feeding ants in several countries in northern South America. The ant tribe Atini contains 250 described species and is now known only from the Western Hemisphere. They are divided into 4 distinct types: lower agriculture, yeast agriculture, higher agriculture, and leaf-cutting ants. Invertebrate symbionts include 7 families of arachnids. 1 spider rides on the backs of ants. Mites are known to feed on fungus and ant brood. Diapriid wasps are parasitoids, undetected as larvae, but adults must leave very fast. Forehead flies have been detected in nests. Various other invertebrates have been seen, including beetles, crickets, lepidopter larvae, nematodes, and worms. There has been only 1 case of snakes depositing eggs in ant nests in lower agriculture ants. Fungus-feeding ants are clearly a fascinating subject, but require more attention by more researchers. After the lecture and audience questions, an official ESW mug was presented to the speaker before the meeting was adjourned at 8:35 p.m.
Thank you. Does anybody have additions or corrections to the minutes? Can I have a motion to have the minutes approved as read?
So moved.
Second. All in favor?
Aye.
Thank you very much. The minutes are approved. So let's move to officer reports. My dear officers here are on Zoom if you have a report. Okay, so let's move to the introductions of new members, starting with membership secretary, Dr. Matt Buffington.
Thank you. Thank you. Good evening, everyone. So I thought I did something wrong with the export of the data, but it appears we're up to 253 members. Pretty excited about that. And there was quite a few new members since the last regular meeting. That's how I filter this. And I filter out renewing members from new members. The following are new members. Benjamin Burgunder of Owing Mills, interested in medical entomology. Karen Burkhart of Hyattsville, interested in plant-insect interactions. Clayton Zimke of Arvada, not sure where Arvada is. Interested in physiology and natural history.
Arvada is in Colorado.
It's Colorado? Okay.
West of Denver.
A good friend of ours, Mark Hoddle from UC Riverside, interested in the biological control and invasive thrips from UC Riverside. Courtney Painter of Oak Harbor, no listed interest. Jim Woolley of Bryan, Texas, my advisor for my master's thesis, interested in biocontrol. I think he's been a member before, so he's sort of a renewing member, but it's been a while. Roy Beckenmeyer of Wichita, Kansas, interested in evolution, taxonomy, paleoentomology, odonades, diptera, acilids, and Hymenoptera apoidea. Raymond Sherbill of Potomac, Maryland, is interested in assisting the organization. And I have on good authority, it missed my list, but Dr. Frank Krell, new member of the INSOC of Washington.
That's about how it goes.
Yeah, okay. Somehow it filtered off my list, but those are all, according to our statistics, those are new members. So it's great to see. Very excited.
Yeah.
Thank you.
Thank you all new members and welcome. Is there anybody new here that would like to introduce themselves? Okay. Is there any unfinished business from the last meeting that you recall and you like to talk about right now? I'd like to check if Warren got my email and read it. Warren, can you hear me and can you let me know?
There.
Mm-hmm.
And Jill, maybe we can move it to the end of the meeting or we can check that
Yes.
and then,
Oh, there you go.
and then wait a little bit.
Okay, so we're gonna go back to new business at the end of the meeting.
Yes.
Thank you.
The business meeting.
Let's move to presentation of notes and exhibit of specimens. If you have something to show us, I'd like to ask you to come forward close to the microphone so everybody can hear and see it, please.
Okay, so maybe it was the December meeting. I couldn't make it in person. I can't remember what we were doing, but were we in Texas? No, I can't remember. Anyway, couldn't make it in. I mentioned that we had received, Loris and I had received from a colleague's widow out in California, some original C.V. Riley books, as she described them.
Oh.
This is one of them. The second one is in such bad shape, I didn't want to bring it. It needs to be rebound. They're not like original books by C.V. Riley. Someone bound all of his reprints. And there's so many bizarre things in here. It's withdrawn from Bowdoin College Library. Pamphlets on Entomology by C.V. Riley. So someone just kind of compiled them, but it's a precious volume. And you can see by the edge here how there's just all kinds of different pieces of paper. There's things that are folded in here. It's very cool. I'm going to invite everyone to come see it up here instead of passing it around, and make sure you got your pizza off your hands. Like, let's keep it, and it's lasted this long. The other volume is like just completely falling apart. So I'll put it over here for, Dawn was supposed to be here. This was like for Dawn. Dawn, if you're out there, this was like for you to kind of take a look, but I'll keep it in my office. So if anyone out there is in the museum and they want to see it, I'll keep this in my office.
Take some pictures of that too.
Yeah, pretty neat artifact.
Has anyone else got something to share with us
Maybe
online?
Dawn. Let me see if I can.
Oh, thank you. Thank you, Matt. I'm sorry I wasn't able to come in. We had a rough day at Beltsville. You may have heard of it.
Yep.
Not the only rough day lately, but thanks, Matt. I'll look forward to visiting you in person and looking at those.
Cool.
Yes.
There are some reprints from C.V. Riley and the Leptanillinae reprint room.
Oh, nice.
What is that?
Yeah. I'll ask for Warren and Jill on Zoom again. Can you hear me? Okay. Warren, did you get a chance to see my email? I just want to get confirmation with you before we move forward on that subject. So under new business, I'd like to note that we currently has a vacancy for an honorary member. And the executive committee is nominating Warren Steiner to fill this honorary member vacancy. Warren is a highly respected entomologist with decades of impactful work in systematics, collections, and curation, and a long history of service to the Entomological Society of Washington, including serving as president in 1998. His contributions have had lasting influence on both the discipline of entomology and on the society. So, is there a motion on the floor to nominate Warren Steiner as a honorary member?
No.
Okay, do we have a second?
Second.
Okay, I'm gonna move forward. Thank you. We have the nomination, and we now can proceed to vote.
He has said nothing, so I'm gonna say he accepts.
Okay. So we need 2/3 of vote from the membership to approval. All those in favor of electing Warren Steiner as a honorary member of the Entomological Society of Washington, please say aye.
Aye.
Does anybody wants to say aye? On Zoom? Did Warren said aye?
He hasn't said that,
Okay,
but he's there.
he's there. So thank you, Warren, to all you've done to the society, and you are now our newest honorary member. So we're ready to move for the presentation for the evening, and I would like to ask Dr. Matt Buffington to introduce our speaker.
Hi, everyone, again. It's my pleasure to introduce Dr. Zach Griebenow for our speaker this evening. Zach got his PhD under the incredibly prolific Phil Ward at UC Davis in ant systematics. He's currently a postdoc in the Hymenoptera unit with the Systematic Entomology Lab. Dr. Gates is his supervisor, conducting Hymenoptera and taxonomy and identification. The talk tonight will summarize his dissertation research in the Ward Lab, focusing on the phylogeny and evolution of the ant subfamily Leptanillinae. And I met Zach probably 6-7 years ago. I think it was at an ESA meeting, and I was struck by, there's just people that you remember. Brendan Boudinot was another one that just kind of locked in my head, also a Ward student. And I've come to enjoy conversations with Zach, and I think you'll find out why when he gives his presentation. So, Zach, the floor is yours.
Thank you very much, Matt. May I have presenter view? How do I, okay. Good. All right. Thank you, everyone. My apologies for that disruption. Thank you for attending my talk tonight on my integrative systematic revision of the ant subfamily Leptanillinae. By integrative, I mean based upon both morphological and molecular data. I want to emphasize that this project does not pertain to my current position within the USDA. It is a summary, for the most part, of research that I have already done as part of my PhD at the University of California, Davis. The contents of my talk are as follows. First, I'll introduce what leptanillines are as organisms, and then summarize the project itself, beginning with the resolution of parallel taxonomy in the Leptanillinae. Then moving on to reciprocal illumination of leptanilline phylogeny from both molecular and morphological data. And then finally, an exploration of male genital skeletal musculature in the Leptanillinae, and concluding with potential future directions for research in the Leptanillinae. So, what are leptanilline ants? At this point, I think we're all pretty familiar with ants. They are numerically dominant and ecologically dominant in almost every terrestrial ecosystem. And in fact, here at ESW, we had a talk pertaining to ants just last month from Catherine Malinsky, pertaining to fungus farming ants in the subtribe Atina. Leptanillines are about as different from the Atina as an ant can get. I like to introduce them by saying that they are to other ants as monotremes are to other mammals. They show a panoply of very derived traits within the formicity, and at least one morphological trait that is uniquely ancestral among extant ants. Moreover, they are sister to nearly all other living ants, which largely belong to a clade informally termed the Euformicea. That ancestral morphological character, uniquely ancestral morphological character that I referred to earlier, is the lack of a distinct basal margin on the mandible, which is shown here as present in the Euformicea. There are no leptanilline ants in the Americas. They are restricted to the Eastern Hemisphere, mostly in subtropical and tropical environments. Workers are exclusively subterranean and so are almost never observed alive. So we know little directly about their behavior. But what we do know indicates that they are specialized predators of geophilomorph centipedes, with colonies moving from place to place in pursuit of their centipede prey rather than forming permanent nests. I should point out some major differences in the morphology of the queen between the two biggest clades within the Leptanillinae, Protonella and Leptanilla. In Protonella, the queen is fully winged, has fully functional compound eyes, and otherwise looks fairly similar to a worker. Workers, of course, these being ants, are all female as well. This is the ancestral condition for ants. There's nothing strange about this, but in Leptanilla, the queen is completely wingless, blind, and is modified in a number of other peculiar ways. These differences in queen morphology have important behavioral and evolutionary implications that will come up later on in this talk. Queens of Leptanilla also engage in cyclical brood production, which means they look like this from time to time. And this, paired with the nomadic behavior of colonies, means that overall Leptanilla biologically resemble the so-called army ants of the subfamily Doralinae. They're almost kind of like mini doralines in appearance, but not in terms of phylogeny. Leptanilline queens also engage in larval hemolymph feeding. In fact, if I recall correctly, they do this exclusively. It's their only source of food. They drink the blood of their own offspring, more or less. This is unusual, but not unique in ants. It's quite similar to the so-called Dracula ants of the unrelated subfamily Amblyopanainae, But in the Leptanillinae, it is so specialized that in Leptanilla, it is so specialized that the larva has a duct that non-traumatically exudes hemolymph for the queen to feed on. Speaking of the larvae, they tend to be long and noodly with tiny heads. Leptanilla take this to an extreme. Leptanilla are also unique among larval ants. Oh, whoopsie. I made the mistake I expected I would make. Sorry. There's a larval Leptanilla. These are unique among larval ants in that the mandibles are exodont, meaning that the teeth face outwards rather than inwards. It's not entirely clear what the functional reason, so to speak, or adaptive reason for this is. Also, the larvae of Leptanilla have a prothoracic process. This is sometimes very elaborate, as you can see in profile view. This is used as a handle for workers during colony movement. So, a potential common name for the Leptanillinae that I considered was worm ant. There is no need for a common name, in my opinion. I'm a systematist, so I really don't see the use, but the thought did occur to me, and I unfortunately did relate it to my advisor. This is an approximation of the face that he made, so I never introduced that common name into the literature. So, worker morphology is quite homogeneous across the Leptanillinae, at least among the major clades. There are some oddballs, like Leptanilla leventa, which shows extreme elongation of pretty much every part of its body. This is an adaptation relative or compared to its closest relatives for living in pre-existing subterranean cavities rather than digging through the soil, as leptanilline workers usually do. In this undescribed species of Protonilla, there are 2 ranks of denticles on the mandible, and not to be outdone, there is a closely related species that has 3 ranks of denticles on the mandible. Then there's Protonilla izanagi, which has 1 rank of denticles, but then multiple rows of tusk-like traction chaetae. You may very well ask, what is this ant feeding on? We do not know. Compared to the workers, male morphology shows tremendous diversity in the Leptanillinae. In fact, the subfamily is mostly known from male specimens, which is the opposite of the case in most ants. This is because in ants in general, including presumably the Leptanillinae, worker ants are much longer lived than males. Males are only produced seasonally and are also less abundant. But the workers in Leptanillinae are exclusively subterranean. Males have wings, so we're much more likely to collect them. Some examples of the weird things morphologically that male leptanillines do. In most, but not all males within the Leptanillinae species group, the mandible is fused to the head capsule, as shown on the top here, rather than being fully articulated. This is nearly unique among male ants, but there are a few cases that have independently evolved this. What is unique among male ants is a feature of the Leptanilla nagefala species group, in which the probasitarsal comb, which is a row of cuticular processes, has seemingly been replicated onto the protibia. This seems to be associated with grasping functionality of the male forelegs. And as far as I know, this is unique among the Hymenoptera. In fact, grasping forelegs have evolved at least 4 separate times in male Leptanillinae, which isn't a lot, but it's strange because it has never evolved anywhere else in male ants, so far as I know. And then lastly, of course, we have the infamous male genitalia, which are often bizarre. You have the usual set of appendages, plus some other things, modified in some very peculiar ways. You might suspect that I'm showing this picture because it's a typical set of male Leptanillinae genitalia, but that's not true. There is no such thing. Male Leptanillinae genitalia are much too diverse for one to fall prey to essentialism when describing them, even by accident. Lastly, there's an important methodological challenge here, which is that Leptanillines in general, and especially the males, are extremely tiny, at least by ant standards. The male specimen shown here is probably one of the smallest ants that exists on this planet, if not maybe the smallest. Here it is shown relative to the world's largest bacterium. So for this and other reasons, my colleague Dave General said that I worked on the Leptanillinae because I lost a bet. Males and females look radically different, which is not anything strange for ants. In fact, that is the default, but they are almost never collected in association, and males are more often collected than the workers, meaning that we can't ignore them from a taxonomic perspective, as is usually the case in systematic myrmecology. So with these conditions, what happened was two parallel and dissociated classifications, one based on males and the other on females, not connected with each other. To resolve this, I needed a phylogeny, including both male and worker specimens. My research objective with that phylogeny is to delimit higher taxa and the Leptanillinae that are reciprocally monophyletic, that is, they are clades, natural groupings, but also uniformly diagnosable based upon both males and workers, because although most of the Leptanilline specimens that we have are male, most of the described species are known only from workers and or queens. So first off, how did I go about resolving this parallel taxonomy? To further emphasize the condition that Leptanilline classification was in prior to starting my research, we had 3 genera known only from females, 4 genera known only from males, and only 1 genus in which both males and workers were both known. By the time I was finished, we had 3 genera: Opimyrma, Protonilla, and Leptanilla. And male and female are by now known for at least some examples of all 3 of these, although I was not involved with Opimyrma in this case, but I did contribute a lot to the other 2 genera. So I talked about inferring a phylogeny. What did I infer the phylogeny from? I used ultra-conserved elements, UCEs, which are genes that are so named for a property that makes them easy to acquire and sequence. In this case, with the HeimV2 RNA probe set. These properties are not relevant to the Leptanillinae in particular, so I won't elaborate on them anymore here. The important thing to remember is that when I inferred phylogenies from UCEs, I was using scores or hundreds of them rather than just handfuls. So in terms of associating workers with clades that were known only from males, first up, we have the genus Leptanilla, shown here in orange, known only from males. With UCEs, with phylogenetic inference from UCEs, we discovered that the species Leptanilla tai, known only from workers, is nested well within Leptanilla. Moving on to Nunilla, another major clade known only from male specimens, shown here in blue. I'll make a quick biogeographical digression here. This marking corresponds to the Patani-Kangar line, which is an important biogeographical boundary in Southeast Asia. Between, well, it roughly corresponds to the transition from deciduous tropical forest in the north to evergreen tropical forest in the south. Now, leptanillines, as far as we know, are not really interacting with the surface environment much of the time, so it's not really clear how this would directly influence them, but based on the geographical distribution of the clades shown here and also some others, it appears that it does somehow, albeit indirectly. It turns out that the species Leptanilla havillandii, known only from workers, was recovered with strong statistical support as sister to the male-based genus Nunilla. So obviously the boundaries of Leptanilla needed some revision. Both Yavanella, shown here in orange, and Nunilla belong to a broader clade that could be called Leptanilla sensu-latu, that is Leptanilla in the broadest possible sense, with the type species of Leptanilla being located in the phylogeny over here. And that type species anchors our generic concept of what Leptanilla is. And Nunilla is more closely related to that type species than it is to Yavanella. So recall how I mentioned that I wanted to have higher taxa in the Leptanillinae that are uniformly diagnosable based upon both males and workers. Well, with respect to males, we can easily tell apart the 5 major clades within Leptanilla sensu-latu, as shown here, using a multitude of male morphological characters. But it is more difficult with the workers, which, as I mentioned before, are very morphologically conservative. One of the main areas of morphological variation is the clipial margin, with workers of Yavanella, so far as is known, having this bifurcating anterior process, which is of unknown function. But Leptanilla havillandii, which appears to be a worker of Nunilla, has this too, so that's not any help in distinguishing them. In fact, the overall habitus of Leptanilla tai and Leptanilla havillandii is quite similar, to the point that at least one taxonomist thought that they might be variations of the same species, although we now know that is obviously not the case based on molecular data. There is some slight differentiation in the lower margin of the petiole, but that's subtle and a continuous rather than discrete character. What is a discrete character is the sculpturation around the torulus. What a torulus is not important. What's important to know is that Leptanilla tai has this waffle-like texture around the torulus, whereas havillandii does not, at least in all of the specimens that I had available. The difficulty here is that this is a feature that is only visible with scanning electron microscopy, which most of us do not have access to. So, since we require a scanning electron microscope to consistently tell these 2 clades apart based upon the worker, I decided to treat all 5 of the clades within Leptanilla sensu-latu as informal species groups. I synonymized them with Leptanilla. There are 2 major clades within Leptanilla for which the worker still remains unknown. I won't say much about associating males and workers in Protonilla, except to briefly talk about Protonilla izanagi, which you were introduced to earlier. Known from both workers and queens, Protonilla izanagi has been collected in temperate parts of Japan, sometimes cold temperate. Phylogenomic inference from UCEs determined that a single male specimen, shown here, which was also bizarre in its own way, a specimen of Protonilla, was a close relative of Protonilla izanagi, but unexpectedly, this specimen hails from central Thailand. To make a climatic analogy to North America, this is a bit like having a lineage of insects for which the only known specimens are from either southern Michigan or the Yucatan Peninsula in Mexico. Now, this disjunction is assuredly an artifact of collection bias, and presumably close relatives of Proteinilla izanagi exist all throughout Eastern Asia. They just haven't been collected yet for whatever reason. The reason is that collecting Leptanillinae workers is backbreaking work. So, UCEs are very useful, not just for inferring the phylogeny of the Leptanillinae directly, but especially for associating dissociated workers and males. But UCE data cannot be acquired from some specimens. Perhaps the specimens are too old, or the specimens are too rare for this to be worth trying, because even non-destructive DNA extraction methods are actually quite destructive when applied to something as tiny as a Leptanillinae. One example of this would be the monotypic genus Pholomyrma, known only from 2 specimens collected in 1907. Since UCEs were unavailable for this lineage, I used Bayesian total evidence phylogenetic inference, relying on both genes and morphological data to infer its phylogenetic placement. It turned out to belong within the Leptanilla revelliarii species group. However, this could be readily argued just by qualitative assessment of the morphology of this species shown here. It conforms to the male diagnosis for the Leptanilla revelliarii species group. But there are some cases of Leptanillinae lineages known only from males for which UCEs are unavailable, in which the male morphology is so bizarre that their phylogenetic position cannot be intuited from male morphology alone. 2 examples here, 2 more examples here. I will emphasize Leptanilla anomala on the left in particular. This was originally described by monotypy in the genus Cyphodon by Charles Bruce, who placed it as Hymenoptera incertae. He speculated on its affinities to the Bathylidae, Braconidae, and the Proctotrupoidea, never once suspecting that it was an ant. This is an understandable mistake because he was under the impression that the specimens he was looking at were female. Brendan Boudinot correctly identified this as a male ant and moreover as a Leptanilline, and I synonymized Cyphodon with Leptanilla based on multiple shared derived morphological traits with the former genus Nunilla that I referenced earlier. However, I did want to resolve the phylogenetic positions of the 4 oddball taxa known only from male specimens that I showed to you within a probabilistic statistical framework rather than just trying to guess where they went. To do this, I inferred their phylogenetic positions from, on the one hand, 64 discrete male morphological characters coded in a binary non-additive fashion, and an alignment of 58 UCEs that were filtered down from a larger alignment so that they would be computationally tractable for the software that I was using. And then I imposed a backbone topology for the analysis consisting of 72 terminals for which both data sets are known. All this means is that they were not allowed to move in terms of their phylogenetic position. For this analysis, Bayesian total evidence inference, I used RevBayes and left molecular and morphological data sets unpartitioned. And all 4 of the morphology-only terminals that I showed to you were included in a single analysis. I wanted their respective phylogenetic positions to inform each other. And they were allowed to move freely in phylogenetic position across that backbone tree. So, in the end, Leptanilla paloensis and Leptanilla CIO1 could not be confidently placed with the data that I had and under the model that I was using. Leptanilla paloensis, incidentally, is known only from a single specimen, which is deposited in this museum. It unfortunately is now missing its head, which is not my fault, by the way. However, Leptanilla anomala is confidently recovered within the Leptanilla havillandii species group, along with the former Nunilla, as we expected. So that's reassuring. Leptanillinae is recovered as sister to an undescribed morphospecies called Leptanilla TH07 with only moderate Bayesian posterior probability. This is within the Leptanilla bethyloides species group. This placement doesn't really make much morphological sense. And I think that this may be an artifact of a phenomenon called bare branch attraction, wherein an aberrant terminal for which only morphology is known, in this case, Leptanillinae, is artifactually attracted to a terminal for which both molecular and morphological data are known, but the morphological data are patchy. And as you can see, the male genitalia are conspicuously missing from this, the only available specimen of Leptanilla TH07. So I was not able to code any morphological characters from the genitalia, whereas I was able to do that for pretty much all of the other male representatives included in this analysis. So within Bayesian total evidence inference, we were presented with a range of analytical scenarios. There's cases in which the phylogenetic position of terminals known only from male morphology is just unresolvable with the data and the model that we used. Cases in which it is confidently resolved in the case of Leptanilla anomala, and also a case in which the inferred phylogenetic position appears to have been misled by analytical artifacts. Now, this is the point where I was going to talk at some length about all of the bioinformatic contortions that I went through in order to ensure that my phylogeny of the Leptanillinae was as robust as possible to perturbations in the data and the model being used. This would have been extremely dull, so I'm not going to do that. Instead, we're going to talk about male genitalia. So male genitalia in insects, as Ulrika Aspuk put it, are a favorite toy of evolution, an everlasting temptation to morphologists. And evolution has toyed a great deal with male genitalia in the Leptanillinae. They are of interest not just for elucidating evolutionary relationships among the Leptanillinae, but the anatomy of male leptanilline genitalia is interesting in its own right for functional and evolutionary reasons. The diversity of male leptanilline genitalia and the weirdness of the modifications were often superlatively alluded to in myrmecological literature, but no one had actually described it in any detail. So Ziv Lieberman and some other colleagues and I set out on a comparative study of male leptanilline genitalia compared to examples in other ants. And if you want to read about this in more detail, this is the paper. So, male leptanilines are too rare for mangling them by manual dissection to be a strategically feasible option. Instead, we used microcomputed tomography for virtual dissection of leptanilline genitalia, which is to say that we made three-dimensional reconstructions of the skeleton and the musculature from image stacks, usually generated with X-rays. These scans were obtained at the following institutions under a wide variety of different conditions. So, in order to understand just how strange the male genitalia get in the Leptanillinae, I need to briefly summarize what they usually look like in Hymenoptera. The terminology that I will be using here is homology explicit and is grounded in the phylogeny of the insects as a whole and comparison to other insects. In the Hymenoptera, first off, for the male genitalia, at the base, we have a usually ring-like structure called the cupula. Distal to the cupula, we have paired appendages called the gonopodides. They are usually articulated into a proximal gonococcity and a distal gonostylus. Medial to that, we have the vulselli, Latin for little tweezer. These consist of a lateral paraciculus and a medial lateropenite. As you can sort of infer here, they're usually opposable. And then in the middle, we have the penial sclerites, which are what actually convey the spermatozoa into the female genital tract. Aside from the genitalia themselves, there are a number of abdominal sclerites that are closely integrated with those genitalia. We have abdominal sternite 8, abdominal sternite 9, and abdominal tergite 9, 10, and 11, which are sort of enclose the male genital capsule. This is all a bit abstract, so, well, here's a diagram, sorry. This is what it looks like in situ with what is a very conventional ant in terms of male morphology, Myrmica ruginotus. So we did complete male genital skeletal muscular reconstructions for 9 representatives from across the Leptanillinae, including almost all of the major clades for which males are known, compared with 3 representatives from outside the Leptanillinae in the Euphormisia. These are then related to each other in this figure by a cladogram onto which are mapped scleritic transformations in the male genitalia. And as you can see from the diagrams here, something has gone horribly wrong in the Leptanillinae. To summarize what has horribly gone wrong, there is a trend towards extreme skeletal muscular simplification in male genitalia in the Leptanillinae. This does not seem to be necessarily correlated with miniaturization. There are many hymenopterans that are as small or smaller than the Leptanillinae, in which the male genitalia are less simplified than what we see here. As an example, looking at the vulselli, ancestrally in leptanolines, the paraciculus and lateropenite were articulated, acting somewhat like a thumb and forefinger. In Protonilla, the paraciculus and lateropenite have been fused. They're no longer articulate, but they are still distinct. In Leptanilla, they are now completely indistinct. And in the Leptanilla nodulifala species group, the vulselli have been fused at the base to form a gigantic wishbone-like structure. This is unique in ants, but there are parallels in other hymenopterans. Taking things even further, in 2 different clades, the vulselli are lost entirely, as seen here. The loss of the vulselli has evolved 2 different times in the Leptanillinae, which is pretty remarkable because, as far as I know, this has never occurred in any other hymenoptera. Another example of general male weirdness would be abdominal sternite 9, which looks like this in most ants. There's an anterior spike called a spiculum, which serves as the attachment point for the sternocoxal muscles that connect the genital capsule to the rest of the abdomen. In Protonilla, abdominal sternite 9 looks much as it does in other ants, nothing strange happening here. But in Leptanilla, it goes completely off the rails. In the Leptanilla Thai species group, abdominal sternite 9 appears to have been completely lost. There's no trace of it left. Fusion to the gonococcides has happened twice, and also fusion to gonococcides and abdominal sternite 8 has also happened twice. All of the modifications that I've listed here are unique in the Hymenoptera, as far as we know. In the Leptanilla nodulifala species group, the abdominal sternite 9 has been prolonged into whisker-like appendages that I termed mulciators, because the phrase posterolateral filiform processes of abdominal sternite 9 does not roll off the tongue, and there's nothing like this in any other Hymenoptera. So to show you a case of what this actually what these kinds of modifications to abdominal sternite 9 actually look like in situ. Here's abdominal sternite 9. In this case, it is fused to the gonococcydes, but I've sectioned it off, shown by the dotted line. Here it is, again, in ventral view with the gonococcydes sectioned off with abdominal sternite 8 and abdominal tergite 9 and the associated muscles. Here it is fused to the rest of the gonococcydes. If you flip this upside down, voila. You might be wondering how this thing flies. I don't know. So this is exceptional. The male genitalia are ridiculously diverse in the Leptanillinae, but the diversity is not without parallels in the ants. Other somewhat comparable cases include, Leptomyrmex, the so-called spider ants, at least the so-called Macroleptomyrmex clade within Leptomyrmex, and Diachema, and also the army ants of the Doralinae. What is the commonality among these lineages? Well, that would be flightless female reproductives. In the Macroleptomyrmex clade, we have a flightless worker-like queen. In Diachema, we have no queens whatsoever. Instead, we have reproductive workers called gamergates. That's gamergate, not gamergate. And then in the Doralinae, we have wingless enlarged queens, as seen here, which, as I alluded to earlier, are kind of like gigantic Leptanillinae queens in appearance. So derivation and disparity in male ant genitalia are correlated with queen winglessness, and that is certainly the case within the Leptanillinae. Queen winglessness evolved here. This is Leptanilla, which contains by far the majority of morphological diversity within the Leptanillinae. There's nothing especially surprising about this, although the evolutionary causes have not yet been robustly scrutinized. However, what is a bit more surprising are the patterns of male genital diversity when comparing different clades of Leptanilla to each other. So the two most diverse clades within Leptanilla are the Leptanillinae species group and Leptanilla reveliarii species group. Yes, they are highly derived in terms of their male genitalia, but what you see when you compare different species is more or less variations on a theme. Conversely, the Leptanilla nagifala species group and Leptanilla havilandi species group are very species poor. As far as I can tell, this is not collection bias. They just genuinely are more depauperate. But at least qualitatively, male morphology is much more varied within these two clades. So why the greater morphological disparity in these two? I have the beginnings of a hypothesis, and to understand it, we have to turn back to the abdominal sclerites that are associated with the male genitalia. From abdominal sternite 9, we have sternocoxal muscles that attach to the cupula. These are the only muscles that attach, at least in ants, attach the genital capsule to the rest of the body. And then the cupula is attached to the remainder of the genitalia by the turgocoxal muscles. This is the only attachment for the cupula to those other distal sclerites. So in the Leptanillinae, there have been multiple occasions where these connections have been lost. Either the turgocoxal muscles become intrinsic to the cupula, meaning they just don't go anywhere. They originate inside the cupula, they stop inside the cupula, which is really weird to look at. They're lost, and often the cupula is completely lost, or it's fused to the rest of the genital sclerites. In the end, the male genitalia in most Leptanilla, not all of them, but most of them, are no longer connected to the body by cupular musculature. So within the Leptanillinae species group and Leptanilla reveliarii species group, there is simply nothing whatsoever connecting the genitalia to the rest of the body, and they kind of spontaneously fall off. which implies, although we've never observed leptanillines in copula and probably never will, that in these two clades, males engage in suicidal mating, which is a consequence of intense sexual selective pressure on the male. It's a very well-documented phenomenon in honeybees. It's a very intense form of male-male competition. Now, comparing these to the Leptanilla nagefala species group, that musculature is still lacking, but abdominal sternite 9 has become fused to the Gonocoxides in both of these clades. That's the pale green area back here. What this means, or at least what I would predict, is that the direct fusion of abdominal sternite 9 to the genitalia, and abdominal sternite 9 is still, as you can see here, connected to the rest of the body, would preclude genital detachment, and thus suicidal mating. If that is the case, then perhaps sexual selective pressure in these two clades has increased on genital morphology itself, rather than sexual selection or male-male competition being mediated through suicidal mating by breaking off one's genitalia and one blocks access of other males to the female. But there's no direct way to test this. It's just a hypothesis. So what do I plan to do in the future, if anything, with the Leptanillinae? Well, I would like to do divergence dating of the Leptanillinae to anchor their phylogeny in geological time and answer the question of why there are no Leptanillinae in the Americas. Their ecological niche in the Americas is filled by Typhlomermex and the Amblyopanaini. To continue the mammalian analogy I made earlier, if the Leptanillinae are spiny echidnas, then the Amblyopanaini are hedgehogs. So in the early Cenozoic, there were two land routes into the Americas from the Eastern Hemisphere, and the climate was very warm at the time. So it is conceivable that the Leptanillinae could have crossed these land bridges, but they didn't. Was it because they originated too late for this to happen, or was there some other reason? This is the kind of question that could be answered with divergence dating. There are no leptanilline fossils, unfortunately, and I'm going to assume that there never will be because the potential for that to happen is extremely low. However, the fossil record of the rest of the Formicidae is quite rich to a degree that I think is not commonly appreciated. So these fossil records can help inform clock models that include the Leptanillinae, if we include both Leptanillinae and lots of outgroups in the same analysis. To do this, I will use an unresolved fossilized birth-death process. By unresolved, that means that the extinct exemplars within the phylogeny are not allowed to move across the tree. I will have 79 extant leptanilline terminals, 72 non-leptanilline ants, and 11 outgroups that are from outside of the ants, plus 162 fossil observations that are constrained by prior knowledge of ant phylogeny. I haven't done much with this yet, but one thing that I have done is to get a prior probability distribution for origin time. I won't bother trying to explain what that is because it would be painful and rather dull, but I've done it. I have inferred an origin time for the clade Uaculeata, which is what encompasses the entirety of the terminals that I'm going to include in this phylogeny. This is a probability distribution expressed in millions of years. You can see there's a big peak here at about 145 million years ago. The origin time is one of the parameters of the fossilized birth-death model. It's the point at which the fossilized birth-death process began in geological time. Still very much an embryonic effort. So lastly, I'd like to thank everyone who loaned or gave me specimens. This project heavily relied on museums because The Leptanillinae are extremely difficult to collect. My advisor, Phil Ward, has been collecting ants worldwide for over 40 years now, I think, and he has only collected Leptanillines twice. I made a very hearty effort and failed. I also want to acknowledge everyone who helped me in other ways, colleagues, co-authors, etc., and of course, thank Phil Ward himself, especially. Before I open the floor for questions, I would also like to nod to the work that I have been doing here for the USDA. If one would like to identify a pine sawfly, look at this QR code. It may be helpful. I hope it will be helpful. And with that, I think we can open things up for questions. Yes, Alyssa.
Be a fairly simple question, but I mean, other than being subterranean, why are they so hard to collect? Are they just super cryptic?
Well, they're tiny.
Okay. Yeah.
That's one thing. They are subterranean in the sense that it's not just that they're underground, it's that they are living in the soil itself. With many cryptic ants, the ants are inhabiting leaf litter, and there are very effective methods, namely Winkler sifting, for collecting leaf litter inhabiting arthropods. Winkler sifting does not work, at least not reliably, for the Leptanillinae because they do not live in leaf litter. As I said, they're in the soil itself. They're very small. They don't make permanent nests either. It's just this colony of extremely tiny ants that are moving from place to place, not even digging tunnels, presumably, just kind of wiggling their way between grains of soil. So the only efficient way to collect the workers is to slam a backhoe into a termite mound, or, well, any kind of digging implement, or to sift soil. That sometimes works quite well. But with the males, since the males are winged, they can be collected with any method that you use to collect flying insects. So they show up in malaise traps quite a bit. Not a lot. I think Leptanillines are still numerically pretty unusual, but that's why we have so many more male specimens than worker specimens. Yes.
So this is a chat question. Actually, I'm going to see if James Robertson can unmute and maybe he can ask the question himself.
Hey, Zach, this is James. First of all, wow, awesome talk. Thank you for that. My question is in regard to their unknown biology. And I'm wondering, has anyone done any gut content analysis on the few specimens that you have, either looking at morphological aspects of what's inside their gut or trying to sequence them?
I don't need to recite this back. Okay. Just making sure. So the answer is no. I don't believe anyone has, because there are so few fresh specimens to work with. There have been studies of the glandular complements of worker specimens in the Leptanillinae, which is unrelated, but that's the only histological research that I'm aware of that has been done. So no direct research on gut contents, no isotopic analysis, because we do know what they're eating with a fair amount of certainty. I've seen them doing this myself. They are very much specialized predators on Geophilomorph centipedes. In laboratory settings, they do sometimes feed on other things, but I'm not sure how realistic that is in the wild. There is one species of Protonella that, when given the choice, refused centipedes and instead fed on forceps tails, which are the family Gepigidae in the Diplura, but that's an exception to the rule. So no, this has not been done yet. Matt?
Can you describe this suicidal male situation?
Okay, so
what's suicidal about it?
So the question is, describe the suicidal male situation and what is suicidal about having your genitalia ripped out of your body. So at least in insects in which this behavior has been observed, it is lethal for the male. So the most famous example would be the honeybee, hopping the drone, as it's called. So the genitalia are ripped out of the body, and then the male dies shortly thereafter, I assume, just due to shock. I'm not familiar with insect physiology, so I don't really know, but I'm assuming that it is extremely bad health-wise for a male leptanilline when the genital capsule just falls off of his body. And also, bear in mind that leptanilline males have extremely vestigial mandibles. That's a shared derived trait of the subfamily, so they're not really doing much feeding of any kind. So presumably they are living for an extremely brief period of time. By analogy with other ants, they're probably dead anyway shortly after mating, even if they're not engaging in suicidal mating.
So following that logic, what is the selective advantage to having what appears to be deciduous genitalia? Does it block it?
Yes.
It plugs the female.
Yes. So the selective advantage, presumably, of engaging in suicidal mating is that the male is blocking off the access of other males to the female because there's this large object already there in the way. That would be the advantage.
So it locks in.
Yes.
Okay.
Yes, in honeybees, it kind of inflates because honeybee genitalia, not many people appreciate this, but they're very bizarre in their own way. People think, I mean, I think of honeybees as being the most boring insect in existence, but the male genitalia are very derived in ways that would be unthinkable for a leptanilline. I won't get into the details, but what they do is they kind of inflate and then lock in, as Matt put it, and then the whole thing just pops off. It should be pointed out that in Apis mellifera, the honeybee, their suicidal mating does occur, but there's still a single pair of muscles that is holding the male genitalia in place. So it's conceivable that even male leptanilines that still have an attachment, a muscular attachment of their genital capsule to the rest of their body, that maybe those are engaged in suicidal mating anyway. It's just that it's pretty obvious that that's what's going on in those lineages where it's not connected by any connective tissue whatsoever. It's falling off. Presumably that's what's going on here. And because suicidal mating is a form of male-male competition, it's presumably being driven by sexual selection. When it's not occurring, therefore my hypothesis is that sexual selection starts to act on more parts of the genitalia themselves. Now, the question of why wingless female reproductives in ants are associated with apparently increased rates of male genital diversification is a very interesting research question, but not one that I had the time or ability to answer comprehensively. But I hope that someone will do that in the future. Yes.
So I have a question from the chat. I'm going to see if I could be loud enough that the mic can pick me up. It does sound like it online. So great talk. This is Sosha Usmani. Great talk. Were many of these species defined before molecular tools, i.e. purely morphological based, or are a good chunk of them relatively newly discovered?
So was that audible to everyone on chat?
Yeah.
Okay, good. So most of the described species of Leptanillinae that currently exist in the literature have not been recently described. I would say I did describe a few myself. Most known species are based just on the worker. The reason for this is that conventionally in systematic myrmecology, the study of ant evolution and classification, we rely mostly on workers, and males are just kind of a supplement to defining species because workers are usually much more accessible. So myrmecologists followed that tendency when describing leptanilline diversity. And The very famed and esteemed myrmecologist Barry Bolton in the early 90s, in the first revision of the Leptanillinae, pointed out the profusion of genus level taxa that were described based only upon males, and he urged everyone to please just stop doing that because it exacerbated this problem of parallel taxonomy. So when I described a new species, I tried to always make sure that there was a worker involved. There were 2 cases where I did describe a species based only upon males, but the reason for that was because those were representatives of 2 clades that were, let's see, hold on, sorry. 2 clades that were known only from males, from male specimens that had not been described whatsoever, and I was going to treat them as informal species groups, one of those being the Leptanilla nagifalo species group, the other being the Leptanilla bethyloides species group. I needed some kind of actual scientific name to anchor or to be so that we could be able to refer to these 2 clades that are still only known from male specimens. That was very long-winded. Did that provide any insight, and is there anything further that I can say to clarify?
Yeah, but we do have another question.
Okay.
John, oh no, we do have it. Yes, I did. Thank you.
You're welcome.
John Gallus says, can injured centipedes be used as some kind of trap to attract bees to move them to the soil? I've seen them used to attract porrid flies to millipedes, for example.
That is a good question. This sort of thing has been attempted actually with a completely unrelated ant clade called Poneracantha. They're much larger than Leptanillinae, but they're still hard to find. They are nocturnal millipede predators, and a way to collect them is to find a millipede and then tie it down and then wait, and then overnight they'll start attacking it. It's kind of sadistic, but so the question being whether we can bait leptanolines with centipedes. In theory, yes, but the problem is that the centipedes are themselves small. They are long, but they are extremely narrow. They're these extremely elongated, wormy centipedes, the order Geophilomorpha. So I don't know how one would go about doing that. And they are exclusively subterranean themselves. There's no way to get around the fact that all of this is happening in pores within the soil. It's very difficult to look at directly, but I wonder if one could, I don't know, come up with some kind of pitfall trap with bait in it. However, I will say that as far as we know, leptanoline ants are active predators, not scavengers. So they would be looking for probably a live, uninjured centipede, because they all kind of, as far as we know, like all gang up on the centipede and then sting it repeatedly and it's paralyzed. And then they bring the larvae over and the larvae attach themselves to the centipede and feed on it. Matt?
Going back to the bare branch attraction, what would happen in your analysis if you excluded the characters for which those terminals lacked the characters that you couldn't code?
So the question was relating to the phenomenon of bare branch attraction. What would happen if I excluded those particular morphological characters that I could not code for Leptanilla TH07 because the only specimen is missing the genitalia? That would be a good thing to do to assess whether this is actually bare branch attraction. If it is, then I would assume, I would guess, that Leptanilla astylina would not show up within the Leptanilla bethyloides species group as sister to Leptanilla TH07. My guess would be that it would go off somewhere else in the phylogeny with very weak support, as did Leptanilla paloensis and Leptanilla CIO1. But that's not something that I considered doing. But that is what one would do to confirm definitively whether this analytical artifact was in fact driving the inferred position of Leptanillinae astylina.
Amazing talk.
Welcome.
Yeah. I think we're getting close to have to rename ourselves the Ant Society of Washington.
So Warren did get back to us and said, thank you all for the wonderful honors. As usual, I got distracted at that surgical moment and missed out, but please know that I am flattered and I have started developing wings.
Oh.
Yay.
And thank you, Zach, for a great talk on this cool group of ants. My teehees are itching.
And thank you all for joining us tonight. Can I have a motion? Oh, yeah, sorry.
Oh, yeah.
It's one of our new things. So can we have a motion to adjourn the meeting? Second?
Second.
All in favor? Motion carries. Thank you very much and good night.